Hi there, I'm Helena Faust, and I'm gonna talk you about antibodies specific to human papillomaviruses, serology, and assays, serological assays. As you see here, I have a picture describing what serum is. So serum is basically from our blood. If we let it to room temperature, all blood cells, they will coagulate. And the liquid, what's left over there, it will be serum, and antibodies gonna float there. So serology, this is science of antibodies. But where are the antibodies coming from? Antibodies are produced by B cells in our lymphs, and they are called plasma cells, those B cells, and these are producing antibodies. Antibodies can be raised against antigen, what's natural, like a human papillomavirus, or artificial, for example, a virus-like particle. And our antibodies, they are in all of our body liquids, for example, blood, saliva, mucosa, and so on. This is a basic structure of antibodies. Antibodies are glycoproteins, and they have heavy chain and light chain. There are many antibody classes too. They can be IgA, IgD, IgE, or IgG, IgM. Most prevalent are actually IgG, what's about 75% of our total IgGs in our body. And IgG is also long-lasting, and it's very prevalent in serum. If we talk about human papillomaviruses, then also IgA is detectable, but it has shorter lifetime, and it's more in secretions and mucosa, and also showing more current infections. Antibodies are very, very specific, and they can bind specific epitopes. This picture here illustrates which kind of conformational epitopes they can bind to. There are characteristics of antibodies, how antibodies are described. For example, specificity, that is, that antibody binds to a certain epitope, for example, linear or conformational. Affinity, that means strength of interaction between antigen and antibody. Avidity shows overall stability of the antigen and antibody complex. And cross-reactivity shows that antibody can actually react with several antigens, particularly if chemical groups are evolutionarily conserved, meaning, for example, in the case of HPV, very close relatives, like HPV 6 and 11. So how can we detect these antibodies? First, if we want to set up an assay, we want to basically, we need to know what we want to see. Meaning, what kind of antigen induces antibodies we want to detect. Secondly, we have to have such kind of antigen for that assay then. And then we have to decide what type of antibody we want to detect. Is it IgA? Is it IgG? Is it neutralizing antibody? And then we have to think how to make it measurable, meaning what kind of secondary antibody to use. It can be colorimetric, light emission. Then you have to consider always cost and specificity and sensitivity matters. And of course, always, if you make a new test, you have to have positive and negative controls. So let's talk a little bit about secondary antibodies. So these we need to detect and enhance the signal. For example, we want to enhance IgG. Then we can choose between polyclonal or monoclonal antibodies. Usually in serology are used monoclonal antibodies because they have a very high specificity and they are recognizing only a single antigen epitope. While actually those antibodies that are floating in our own body against viruses, for example, they are always polyclonal because our immune system tries to cover all different spectrum to protect us. So what is the best antigen to detect the virus-specific antibodies? Of course, most of the assays, the real virus is used. But in HPV, problematic is that we cannot really produce it in such kind of high amounts in culture as a real virus. So therefore, we need alternatives. So therefore, we are using capsomeres or virus-like particles, pseudovirions. So these are structurally similar to real viruses, but they are not real viruses. So here are the L1 monomers, and if 5 monomers come together, it's called capsomeres. And spontaneously, those capsomeres, they make a virus-like particle. So virus-like particle looks very similar to the real virus, but it doesn't have DNA. Or in most of the cases, if VLPs are produced, it's made only out of a major capsid protein L1, and L2 is missing. So here are the so far used antigens in studying HPV antibodies. The most simple is GSTL1, and it's expressed in bacteria, used in seroepidemiology. It's easy to purify and produce such kind of antigen. Level further is virus-like particle out of L1 capsomeres, but I have assembled into particles. These virus-like particles are so far produced almost all organisms like bacteria, yeast, insect cells, mammalian cells, and lately I have heard that even in plant cells. These are used in vaccines, HPV vaccines, and also as antigens in serology and structural studies. Step further, closer to the natural viruses, are pseudovirions. It consists both L1 and L2 proteins plus pseudogenome, what will be a reporter plasmid to be able to monitor infection, for example. These pseudovirions are always only mammalian cell cultures produced. They are used in vaccinology, structure, viral life cycle studies. Step further are quasivirions. So it's like a pseudovirion, but it has a real virus genome. And as you can understand, this is already a biohazard here. And they are very uncommon to use for any other kind of studies than viral life cycles and transformation studies. And then we have native virions, what we actually can collect from warts, for example, but it's always so little amount that you cannot really waste it for any assay. So therefore they are like used for viral life cycle and transformation studies too. And they can be also like grown in organotypic graft cell culture, but again, the yields are low. So you cannot waste it for just detecting antibodies. So why do we need to detect those HPV antibodies at all? Well, one answer here is that the HPV DNA is transient and usually clears within a year. So looking in that population, looking only at DNA, one can think that, oh, it's actually very rare virus. But that's not true because it's just cleared this infection. So if you want to know, has a person been infected, then we need to look at antibodies. We want to know about the spread of the virus infection in population, called seroprevalence. Then antibodies have also some kind of diagnostic values, especially E6 and E7 oncoproteins antibodies to them, but these are still in establishment state and not clinically used. And of course, vaccine research. We have to detect antibodies to HPV to know HPV vaccine-induced antibodies, their levels, durability, and their capacity to neutralize. To detect anti-HPV antibodies, it's a challenge because so far we already know 206 HPV types, but 12 types are established as oncogenic. So another challenge is that not all infected people actually seroconvert because the infection can be in very low copy number, so that immune system even do not respond with antibodies to it. And seroconversion also takes a long time. Like it might take 1 year of infection before antibodies come. And of course, all methods have detection limits. What is noise and what is signals? And have a balance between sensitivity and specificity is always also a key question. Good news here is that HPV-specific IgG antibodies are very stable and they are detectable over decades. So now I give you a slight overview about different HPV serology assays. They can basically be divided into 3. First is neutralization assay, what's a golden standard assay, and it measures protective antibodies, meaning all kind of antibodies, IgG, IgA, IgD, Then it's binding assays. One can call it ELISA, what's specifically detecting, for example, IgG. And then this ELISA can be further developed into high throughput antibody binding assays called multiplexing. And here we have, for example, competitive Luminex assay, TST Luminex assay, and pseudovirion heparin Luminex assays. So let's talk a little bit about what those methods can detect. So this is a figure about antibodies what's detected by induced by, for example, vaccination. And this red circle here can show all of the antibodies which are produced. But as you see, none of the current methods most likely are not detecting them 100%. But this orange circle here, it's ELISA. So this binding assays, ELISA type binding assays can detect the majority of them. Neutralization assay looks subunit of those, and the most narrowest is competitive Luminex assay. So let's talk about golden standard assay, neutralization assay first. This is a cell culture assay. It's rather laborious, low throughput, but very important as it measures protective antibodies. And this is important when we have vaccine studies. So on the left panel here, it shows a situation where sera has no antibodies, no neutralizing antibodies. That means that those pseudovirions carrying reporter can infect those cells, and it can be chemical signal afterwards measured. On the right panel here, there was antibodies. Antibodies were binding pseudovirions, So the virions were blocked, and so they couldn't infect those cells, meaning there was no signal. Binding immunoassays, ELISAs, are also different types. They can be direct or indirect, sandwich assays, and competitive assays. Classical ELISA is just like one HPV type can be detected at a time. For example, one antigen HPV16 is coated in the well, and then step by step will be done detectable this antibodies. This is used in seroepidemiology and also some vaccination follow-ups like serology studies. And historically, it's most widely used method. These days, binding assays are often multiplexed because there are so many HPV types. So it's better to save time and money and material, basically, and detect all those antibodies at the same time, but still specifically. These methods are high throughput and liquid is based bead arrays, and basing on Luminex technology, what is machine what has a two lasers. And I always tell that this is a little bit like a cell sorter working, but instead of sorting and detecting cells, it's sorting little plastic beads. And all those beads have their own identity, so that you can use 100 different beads where are coupled antigen on it at the same time and detect them all. And if in ELISA we had antigen in the bottom of the well, then now we have such kind of tiny beads where the antigen is basically glued. And if you get your results out of the machine, then you get it in median fluorescence intensity units. So what kind of multiplex methods are used right now? One assay was Merck Gardasil study assay, what was competitive assay, meaning like competitive Luminex, but it was competitive in a way that it was multiplexed format. So it's a surrogate for neutralization assay, where was only measured one epitope, one known neutralized epitope, and binding to it. Basically, how it is, that first there is a signal because monoclonal antibody is binded here. If there was serum antibodies, then those serum antibodies, they're competing off this signal, meaning that if it was antibodies, you lost the signal. It was a little bit laborious method, so Merck was also making direct binding multiplex assay for their Gardasil 9 vaccine validation studies, detecting all of the types they have a vaccine about. Only problem here is that, of course, they are using the same antigen in the assay as they are injecting in the people. So, of course, they see antibody binding afterwards for the same antigen they have been injected in the people. But is it also like natural or not? That will time tell. Widely used method also is GSDL1 multiplex immunoassay. As told, this bacterial-derived glutathione detransferase, it's a rather large molecule by itself, is tagged to the protein L1. And it's mainly used in German Cancer Center. It's Dr. Paulita and Waterpower group, when they can detect more than 30 genital HPVs and more than 50 cutaneous HPVs at the moment. Mainly they are using it for seroepidemiology, but they have been started to use also for vaccine evaluation. We are using in our laboratory heparin pseudovirion Luminex assay. It's published since 2010. And what's our setup is that earlier studies have been showing that only correctly folded particles of the virus attached to heparan sulfate proteoglycan. This is rather universal attachment factor for many viruses, so HPV is nothing special here, but still it avoids noise, meaning that just floating L1 capsids or L1 monomers could bind to the bead or to the bottom of the well, meaning that we have more accurate measurement of the antibodies too, because those antibodies which we are measuring then they're specific for the virus-like particle or pseudovirion, not just with the L1 monomer or capsomer. Those pseudovirions are produced in mammalian cell line 293TT. And we have plasmids that expressing both L1 and L2. And from cells, we are gathering pseudovirions by lysing cells and purifying with density gradient ultracentrifugation. At the moment, we have constructs for 17 genital HPVs and also some skin-habiting HPVs and Merkel cell polyomavirus. So validation of the method. One needs for that positive serum panel and negative serum panel. For our positive serum panel, we have proven HPV DNA in cell samples, and we can then estimate that about 50% of them should have also antibody response. Then we have strongly monospecifically reacting serum called international standards. These are established so far to HPV 16 and 18, but still work is ongoing and hopefully we will cover more types soon. When I'm doing routine testing, we are using sera what reacts with wide band of HPVs and also vaccine-induced antibodies from the sera. In our negative panel, we are using children sera, depending of the type. For genital HPVs, children below age 10, and for skin types, children below 5. And then we, according to the negative serum panel, we are using the WHO algorithm to establish cutoff, which is average value of negative panel plus 3 standard deviations. So in the end, pseudovirion Luminex as a modern HPV serology has its good sides, as it has a high throughput capacity. We have validated it with natural virus infection. It has a good correlation with neutralizing antibodies. We are using it for seroepidemiology and vaccinology. So we are studying HPV antibodies for seroepidemiology and vaccinology. As I told earlier, DNA in cervix is transient and therefore doesn't give a full picture. And it's also much easier to have access to serum because, for example, thinking about males do not take part in the protein screening like females. So accessing sera is always a little bit easier. And antibodies to HPV are very stable and represents information about cumulative infection then. And then we are using this information to set up prospective studies, the association between disease and infection. As serum antibodies specific for the infection, this has to be there in person before the disease comes to present. Also, after vaccination, serum antibodies is marker for successful vaccination and also protection after vaccination. And if there was antibodies already before vaccination, we know that the person was infected before. That means that the protection most likely is not as good as in not infected persons. So conclusions for my talk is that the major capsid protein, L1, is the most antigenic protein of human papillomavirus. Therefore, it's mainly used as antigen in all kinds of antibody testing. And the best antigens so far to use in such kind of seroassays are virus-like particles of pseudovirions. Because better the viral particle conformation is, better result you get, better and more reliable. Neutralization assays remains as a golden standard method so far. And all kinds of binding assays, ELISA is reasonable to multiplex as it technologically possible. And in the end, it's an important tool for epidemiology and vaccine research.